let's give a warm welcome to Mr. Faisal as he leads us through the important and fascinating subjects.
So, as explained by Sambo, that I'm based in Durham University. Currently, it's the third year of my PhD. It's a 4-year program, so hopefully next will be the last year. Um, so my primary research topic was based on the supercapacitors, but along with the supercapacitors, I'm also working on water splitting and some other experiments which do include electrochemical analysis and testings. So I'll go through main three important electrochemical techniques, including cyclic voltammograms, which are also known as cyclic voltammetry, CV, galvanostatic charge discharge, and electrochemical impedance spectroscopy. Apart from these three, I'll just allude at the end of the talk about the chronoamperometry and chronopotentiometry, for which applications and for which purposes you can use these two, and why they are helpful in certain applications and why they are not helpful in fields like supercaps and other bits. But the electrochemical techniques are not just restricted to these three, although these are very important, but there are a number of other electrochemical techniques which are very useful in electrochemical biosensors, their characterizations and their evaluation while playing with the materials in that specific field. So I'll just start with the cyclic voltammogram. Before starting to the cyclic voltammogram, so I think we should define what is a technique. So basically, a technique is actually a method or a procedure which we actually utilize to measure, analyze, manipulate, and dig out the electrochemical properties of any material. That is termed as a technique. So when we utilize that technique for certain purposes, for example, for voltammetry. So in voltammetry, I mean the cyclic voltammograms. Um, so we actually run cyclic voltammogram on a specific material, and we get some response as a result of that cycle or that run, and then we analyze that cycle and get some vital information out of it. These cyclic voltammogram, or in general the electrochemical techniques, can be carried out in two different ways. The first one is the three terminal cell. The second way of doing it is the two electrode, which can be done either in this assembly or this. I'll go through each of them on this slide. So in three electrode cell, we actually use one electrode as a working electrode, second is the counter, and third is the reference. And I'm sure you all guys are familiar with it, that what's the role of reference electrode, counter electrode, and working electrode. But I'll just allude that the reference role is to maintain a constant potential between the working and the reference electrode, whereas the counter electrode actually maintain a constant current between these two electrodes. So if reduction is happening at the working, uh, um, oxidation will happen at the counter electrode, and if oxidation is happening at the working electrode, reduction will be happening at the counter electrode, and that's how the current is maintained inside the cell. But that's the three electrode cells, and in real world we don't deal with the three electrodes. So for real world, we need a two electrode in which one is a positive, one is a negative. Um, so in battery research, most often utilized cells are the coin cell assemblies, because uh, they are actually punched with the applied pressure, so that they can couple very appropriately, and they can to minimize the resistance between the electrodes. Apart from these coin cells assemblies, the swedge lock cells are often used nowadays in most of the labs, and this is also a two electrode assembly, and to use it with the electrochemical potential set. So what you do is to combine the reference and counter on one terminal and the working on the second terminal, and that's how you connect it with the
Hello,
electrochemical cell. So
hello,
this is this is another way of doing the
hello.
electrochemical study of the Swedlock cell and getting information on it. Um, but some people
Hello,
do
Mr.
have
Faisal.
the here is the Swagelok cell or assembly, which can also be used to characterize any two electrode materials through electrochemical techniques such as CV, GCD, and EIS. And some labs do have the facility of these stainless steel assemblies, which do have the similar job, which can be done with the Swagelok or the coin cell assemblies. So they are just different in the structuring or in the material, but they do have the same application of these two. So in voltammetry, there are a number of voltammograms or voltammetry technique which can be utilized. The one which is most often used in the electrochemistry is the cyclic voltammogram and the linear sweep voltammetry. There are some other which I haven't touched upon it yet, or the staircase voltammetry, square wave voltammetry, and fast scan voltammetry. Fast scan is somehow now being utilized in the battery field, but I haven't seen it in the supercaps. Maybe in the field of um, magazines can be utilized, but I haven't seen it so far for the supercaps. But even if it is fast, the analysis and the analysis and the data interpretation of this will be exactly similar to the cyclic voltammograms or voltammetry. The so coming towards the electrodes, which are mostly utilized in the three electrodes. Uh, Basically, you use one reference electrode, uh, one counter electrode, and one working electrode. And I've already explained what is the role of working electrode. So this the working electrode is basically the material under the investigation, whatever the material you have developed or produced, and you wanted to explore its electrochemical fingerprints. So you just utilize that electrode as it is. If it is not prepared well, you just follow it through a specific procedure and make it an electrode and use that as it is as a working electrode. Whereas for the reference electrode, you use specific electrode, for example, AgAgCl, mercury oxide, mercury chloride, and standard hydrogen electrode, depending on whatever your application. For example, if your electrolyte is more or less acidic or basic, then you can utilize AgAgCl. But if it is uh, it is actually basic only, then you can utilize mercury oxide, but not the mercury oxide is not always very friendly with the acidic electrolyte. Similarly, the saturated calamine electrodes are also very good in some application like corrosion analysis or the water splitting application. But even if you don't have specific or the required electrodes for any application, you can still convert the potential of that specific reference electrode to any desirable. For example, if you have a AgAgCl electrode and you you measure the potential against it, you can easily convert it uh, into the potential against the reference hydrogen electrode. And there are a lot of protocols on in the literature that how to do it. It's just a matter of some potential, either adding it to the original potential or either subtracting it from the original potential. Then there is another electrode, which is the counter. And for the counter, most of the time we use inert material, for example, platinum or graphite. So why platinum and graphite? Because they have the property to maintain a constant current without degrading or and overcoming the resistance. So that's the main purpose of these two, and they don't they don't facilitate any side reaction during all the electrochemical analysis. So that's why they are preferred, and they're used in the three electrode cells to not distort this cell or the overall property of the working electrode material. Another material which is very often used in electrochemical biosensing application or in the supercaps or in the HER or OER application is the glassy carbon electrode, which is often somehow expensive, but it is reusable. So that's the very good pros of this electrode material, and it can be used for many times if you are dealing it with it properly and polishing it properly until it is discarded. So it can also be used, but the problem with this one is that it requires a very tiny amount of the electrode material, whereas in the real world sometimes we need mass or the material in much more quantity than it required on the classic carbon electrode. So you can still use it in the lab research, but if you are working on an industrial application, so you might not be able to use it. But because in industrial you need a much bigger electrode material rather than a very tiny one. So in terms of convention, so cycle voltammograms can be actually presented in two different ways, whether the potential can start from higher to lower or from lower to higher, and the oxidation and reduction peaks can flip upside down. So why these are two? Because there are two conventions we have. Uh, the first one is the US convention, in which the higher potential is used on the origin and it sweeps towards the low potential. Whereas in the IOPEC one, it starts from the lower potential and sweeps towards the high potentials. I guess most of the literature is based on the IOPEC convention. So you will have the oxidation if it is a redox material, um, oxidation on the top and reduction on the bottom of the cyclic voltammograms. It depends on whatever the convenience for you is to represent the cyclogrammogram. So it's not wrong if you decide to present it in this way or either this way. Once you have any material, let's say I have a material A and I wanted to explore its electrochemical performance, and let's say I run cyclogrammograms and I ended up with a shape of this one which I presented here. So now what's next, and what does it tell me or anyone to anyone who is either very initial or either a very beginner in the field? So first of all, if you get a rectangular shaped CV of any out of any material, so there are two assumptions now. Either your material is purely electric double layer capacitor, which stores charges through forming charges by electrostatic accumulation at the electrode and electrolyte interface. Or another assumption is the material can facilitate some redox activity, and that's how it can be categorized in Faraday pseudo-capacitive material. So that's the first or two assumption or two initial information which I can dig out from the cyclic voltammogram behavior. What else I can do? I can estimate the capacitance of or the capacity depending on whatever the behavior of the material is. If it is pure EDLC or pseudo capacitor, then it is preferred to calculate the capacitance rather than the capacity. And that's why the capacitance should be reported rather than the capacity, because doing a capacity for this material will be somehow misleading, because you're not dealing with any Faraday reactions uh, to deal with the charges rather than the ferrets. So you can actually calculate the area under this whole curve, and using this expression, you can convert it into uh, first into specific capacity and then dividing it by the total potential range, and that's how you can get the capacitance. What else I can still get from this from this one cycle, or whatever I've run multiple cycles? So because this is only one cycle, but I can run it at a multiple scan rates, and the scan rates is depending on your desirable application. If you need very low scan rate, you can start from very low scan and take it up to a higher scan rate, and then you can just estimate the rate performance, that what was the capacitance at the initial scan rate and what was its behavior when you take it up to a very higher scan rate, and that's give you the rate performance, whether or not the material is very good when you are switching the potential very quickly, or whether the material is very good when it is very slow, sweeping the rate between lower to higher potentials. um, when in terms of the battery graded material, so you will have a very distinct oxidation and reduction peak in the cyclic voltammogram. But there will be some, in some material, it is not very often in every material, but in some material you will do have some EDLC contribution, and that tells you that there is still an electrostatic accumulation, but there is a broad Faraday reaction happening at the electrode interface and in the bulk of the material, and that's why it gives you the oxidation and reduction peaks in the cyclic voltammogram. So how can you then idealize that whether or not your material is behaving reversible or irreversible? You can take the ratio of the peaks, peak position or peak current of oxidation and reduction, and just take a ratio. If it is nearly equal to 1, then that means the performance is reversible. Another way of doing it is getting the number of electron involved in a cyclic voltammogram, and then if 57 divided by n millivolt is the exact amount of difference between these two oxidation and reduction peak. If it tells you that the difference is lying around 57 millivolt for electron system, that is a reversible. If it is more less than or more than that, then there is certainly some problem with your system, and you have to actually accommodate or actually identify that problem before going further with that material. So so this is very basic example of inactivated carbon, which is tested at several scan rate, and the reference electrode is used as a AGCL, and you can see the potential ranges from -1 to 0.0 volt. So you can easily get capacitance as a function of the scan rate capability as a function of the scan rates from the real application of the electrode material. The second graph actually shows you the again the 3 electrode assembly in which the potential is recorded as a function of HGO reference electrode, and the material is tested as a function of different scan rates, and then you can play on with the parameters and analysis accordingly. And the third example I've shown here or presented here is actually the potential V. So you can see there is a clear difference between these two, and there is a potential. So whenever you are seeing cyclic voltammogram, and if a potential is shown rather than the potential versus HGO, that clearly means that the testing was carried out in 2 electrode rather than in 3 electrode. If someone is not mentioning AGCL in 3 electrode, I think it will be misleading, because you are now you are contradicting your results or the way of presenting results with the 3 electrode assembly. So this is actually a 2 electrode cell, which is characterized as a function of different scan rates, and then was further analyzed accordingly, whatever you are interested in. So that's the clear difference between 2 electrode and 3 electrode. From whenever you see in the literature that if a potential is presented against some specific reference electrode, that's 3 electrode. If not, that's understood that this is coming from a 2 electrode cell. Coming towards the GCD analysis, so GCD is actually in somehow similar trend. In cyclic voltammogram, we provide potential at a specific scan rate. In GCD, what we do is we provide a specific current, and we set a potential for any electrode material or any cell, or which could be in 3 electrode or 2 electrode, and then that specific current actually reaches up to that potential, and then start discharging, and then again reaching the zero potential from which it started. So this is a whole cycle of charging an electrode material and then discharging an electrode material. This is just one specific example. It is not universal for any material or every material. So you can get different responses in the cyclic voltammogram. For example, the GCD might have some plateaus, and those plateaus actually resemble with the oxidation peak which we saw here. So if your GCD do include some plateaus, proper plateaus, that actually gives you another similar information which you have received from the cyclic voltammogram, but in the GCD it will be in terms of plateaus. So this specific example is actually more or less for either EDLC or the pseudocapacitor material. If your material is purely EDLC, for example, carbonaceous material, and you still have some humps in the charging or the discharging part, that means that you have some functionalization during the activation of the carbon, and that is actually involving in some Faraday activities. That doesn't mean that it is still a pseudo material. It is purely EDLC material, but due to those functional groups, it is behaving like a little deviation from its ideal behavior. But in pseudocapacitor material, it is clear that there are some Faraday reaction happening in the at the interface of the electrode material, and that's why it is differentiated or distinguished from the EDLC material. The axis can be presented either in seconds or hours, because in batteries we don't have seconds. It is a very long time and very prolonged time, so you might get in hours rather than in seconds. The y-axis can be plotted either in potential versus some reference electrode, or if it is two electrode, then it's just a voltage. Once you characterize any electrode or device with the galvanostatic charges in a similar fashion or in a similar manner, you can get the specific capacitance for that specific material by using these two equations. If that material is either a battery graded material, then you can rather stay with the capacity rather than capacitance. The capacity equation would be similar if you eliminate potential from here, and that will gives you the capacity of that material. um, Rate performance can also be obtained from the GCD performance if you do so by running galvanostatic charge as a function of different current densities or different specific current, so you can easily estimate the rate performance. Another very good important or very vital information that you can get from the GCD is the Coulombic efficiency, which is extremely important, because if as a factor of time, the Coulombic efficiency is degrading very quickly. That means your material is losing the ability to store charge very quickly as a function of time, and that can be easily calculated by incorporating the discharge capacitance and the charge capacitance, or the discharge time versus the charge time, and then you can get the Coulombic efficiency. Another last parameter which can be estimated is the potential drop, and this potential drop can give you also give you the value of the resistance, and that resistance can be resembled with the resistance which you can get from the electrochemical impedance spectroscopy. So these are the important bits and bobs you can get from galvanostatic charge characterization. The linear sweep voltammetry, I'll just go through it quickly. So it is also very important. So what is linear sweep voltammetry? It is actually similar to the cyclic voltammogram, but not cycled. It's just a half cycle. Where do we use these characterizations? So normally, if you are running linear sweep voltammetry in a negative potential region, that's where people are using it to explore a material potential for the hydrogen production or H2 production. If you see a linear sweep voltammetry run in a positive potential, that's actually a customized trend or practice to actually explore its potential for the O2 product. But in any way, if you are dealing with one, it produces the second one automatically, because you are splitting the water at one electrode and the O2 is generated. But that's not just the application of the LSV, because I have also seen most of the papers, like people are saying LSV can be done for only Tefl plot, estimation or only the over potential estimation, but I guess that's not the only application of the LSV. If you've read papers on the water and salt electrolyte for the supercaps, in those papers, people have used the LSV as a very important characterization to estimate the actual potential window of the real device. For example, this is the specific example which I've taken it from a published paper. And in this paper, the people have actually done LSV to estimate the actual potential as a function of the molarity of the electrolyte. So which means the LSV is another important characterization that can also tells you the stable working potential of your device without degrading your device by providing it a harsh potential or the current throughout the electrochemical testing. So if you combine both half cycle of the LSV, you should be able to get a full cycloglutammogram shape, and which I just illustrate here to show you that why it is called the half cycle of the cycloglutammogram. So LSV is actually a simple electrochemical technique, and it is similar to cycloglutammogram, but rather it is not a cyclic. It is just a single sweep at a specific scan rate, depending on your application. If whatever your application is, you can decide the scan rate. The scan rate can also be varied. It depends on how fast or how slow you want to interact the electrode material within the electrolyte. I've shown here just a specific example of the platinum wire, which is a very famous electrocatalyst used for the water splitting. And you can see that even if you prolong the potential window, you won't get any cyclic performance. You are just ending up with a single sweep, and then you can utilize that single sweep to dig out the over potential Tefl slope and other bits and bobs from the LSV of the material. So the pros and cons between LSV and CV are that LSV is just a single step, whereas the CV is back and forth. You can easily get the idea of, because LSV won't tell you that which type of material it is, but the CV will definitely tells you that which type of material it is and to which class of material it belongs. The amount of information which you can dig out from the cyclic voltammogram is very high in CV, where it is moderate, or if I can say it is lower in LSV, it will be correct. Cyclic voltammogram are more complex than the LSV because, as I mentioned, if you get a triangular CV, that doesn't mean every time all the time that it is EDLC material. It can be a pseudo material which actually forces you to do further characterization and further analysis before coming to a conclusion that which material it is. So CV is more or less complex. LSV is simple technique. Mechanism involved in is in CV is very it's very firm, whereas in the LSV it is very limited. From kinetics perspective, I think CV can gives you more information on the reversibility rather than the LSV. And in time efficiency, I think LSV requires less time than the time you consume on these cyclic voltammograms to characterize a material thoroughly and estimate what was the actual performance of the material as a function of every scan rate or as a function of single scan rate. From reversibility detection, that's clear that cyclic voltammograms are reversible. You can run number of cycle at a designated scan rate, whereas you have to actually run another experiment for LSV if you want to detect reversibility in through the LSV measurement. The electrochemical impedance spectroscopy, in contrast to these three electrochemical which we have already covered, I think this one is extremely important because it is very complicated, very enriched with a lot of information, and it is sometimes very challenging to actually dig out proper information from it, because it needs some modeling, and not in every case the modeling is possible, because in some cases the modeling is very complicated, that you need some specified or subscribed software which are not usually available to everyone. But still, it is very good technique to tell you very important bits of your electrochemical cell, for example resistances or the behavior of your electrode material or your cell specifically. So this is what you guys might get normally when you deal with the EIS for a certain material. So here is a point A, which gives you the resistance of the electrolyte. So it is most often that if you are dealing with the either water splitting or supercaps or battery, you will utilize the electrolyte, and electrolyte does contribute some resistance to the cell. That resistance is actually coming over here. Then beyond this limit, it comes another region, and this region actually belongs to the electrode material. So if there is a charge transfer resistance between the electrode and the electrolyte ions, then it will show some resistance, and that resistance is regarded as the charge transfer resistance. Then there is another region which comes over here. So if you guys have studied the phenomenon of charge storage in the supercapacitor, that in EDLC material there are a number of layers before the electrolyte form an electrostatic accumulation at the interface. That there are diffusive layer, there are stern layers, and so this is the regime which defines the diffusion of the material inside the diffusion of the ions inside the electrode materials. And then there is a linear region which is almost parallel to the y-axis, and that defines you actually the 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 behavior of the material, whether or not it is capacitive or not. So I'll just start off with a simple example. So if you use an active device like a resistor in a if you connect a resistor with the electrochemical potential set, what you can expect is a single point from that resistor, because that resistor got a single point resistance, and resistors are actually independent of the applied frequencies or the activation frequency you are providing through the electrochemical impedance spectroscopy. Because in electrochemicals, what you do in the EIS is actually providing a range of frequency from a higher frequency towards a lower frequency, and you run that frequency those frequencies at a specific alternating potential. So if you fix that potential and you vary the frequency, then you record or measure the response of that material in response to the sweeping frequencies. So since the resistor doesn't contribute or doesn't affect overall in response to the applied frequencies, it should give you a single point resistance. But since in real world we have some we have a lot of factors when we connect the resistor to the potential state, so we can get a variable on average value. We can get scattered value of the resistor, which if you zoom out, you'll still get a very single point, and that contributes to the total resistance of that resistive material. So I've used here a 10 kilo ohm resistor, and you can see the performance is almost a single point value. But if you zoom it, you can get the scattered value of the resistance value out of the resistor. So when you actually if you want to analyze the electrochemical impedance spectroscopy cells, so there are two ways of presenting the data. One is the Nyquist plot, and another one is the Bode plot. So the Bode plot actually what does it include is the log of frequencies versus the log of impedance, and then you have the phase. Why do we need phase? Because in capacitors and inductors specifically, there is a there is a lag off between the current and the voltage. Similarly, if current is leading, voltage is lagging. If voltage is leading, current is lagging, and that's why it differs within the material, and we'll discuss in the later slide. So since the resistors are independent of the frequencies, you get a single point value. Even if you run number of frequencies, you will what you'll get is a straight line in the Bode plot, because there is no change or no variation in the resistance of that specific resistance. Why is the phase is at zero? Because there is no lagging between current and voltage in the resistor, so that's why the phase is consistently constant throughout the range of frequencies. But what if I use an inductor? Because an inductor is a passive device which store energy in the form of magnetic field, and there is a lagging between current and voltage behavior in this specific material. So you will get zero resistance because these are not the resistive material, and you will get a very a linear or a straight line but parallel to the y-axis on in the Nyquist plot, which is presented here. So this is the very rather typical trend of an inductor if you connect it to the potential state and run EIS measurement. Whereas in the Bode plot, you can see the phase is consistently 90, and that tells you that the potential is leading current by 90 degrees. That is pi by 2, and the impedance value can be a sort of linear line, which tells you when the frequencies are lower, the impedance is lower, and the frequencies are higher, the impedance is higher. And that comes from the relationship between the reactance of the inductor with the inductance and frequency of the applied potential. What will be the scenario in the capacitor is basically, because capacitor is also not a resistive device, it is a passive device. It stores energy in form of charges, which means it builds up potential. So potential will be lagging behind in comparison to the current. So you can get a very straight line or linear behavior parallel to the y-axis on the Nyquist plot, and when you plot it in the Bode plot, so you can get a phase change as a function of the scanning of the frequencies. And you can also see the impedance is high when the frequencies are lower, and impedance is lower when the frequencies are higher. And that's why that's coming from the equation, because the impedance and capacitance and frequencies are inversely related within the impedance or the reactance of the capacitive element. What will be the scenario if we connect resistor and capacitor combine in a cell, and then we opted to do an electrochemical impedance? Because there is now a resistor connected with the capacitor, so we'll have a displacement of the trend towards the lower frequency region, and then it will be again a linear parallel to the y-axis. So that tells you that this combination does have a resistance, but since there is a capacitor, so that's why we are getting a linear region in the lower frequency region. And similarly, you can actually you can study the behavior, how it is changing, the phase is changing when you sweeping from lower frequency towards a higher frequency, and how the impedance is changed from higher towards a lower as a function of frequency. So when you combine resistor and capacitors parallel, everything is changed now, because in previous case we studied that the the trend is actually parallel to the y-axis, and there wasn't any difference. Even if we connect a resistor, that there was just a displacement of the trend, and that's all. But if resistor is connected with the capacitor in parallel, that's how it differs your overall trend. So you get now a proper semicircle formation. And why is that so? Because when you connect them in parallel, it depends on the number of it depends on the value of frequency. So at certain frequency, the capacitor will block the current, and most of the current will go through the resistor, and at certain frequencies current will go through the resistors, and that's how you're getting a semicircle formation. So let's consider these three EIS is a best example of how can we relate it to a real data which you have taken. So for example, you have let's say 4 electrodes starting from K1 up to K4, and you characterize them both through the cyclical timeogram and through the EIS. Now, are they going to actually connect with each other through these two different technique? Because in cyclical timeogram you are providing potential, whereas in the EIS you are scanning actually frequencies. So some people will say no, these are different, but in general perspective they should actually merge together and gives you a final conclusion about the material, whether or not whatever you have dig out from the cyclical timograms they are matching with the information in the EIS. So the first initial guess from these spectra is basically the K1 is very terrible. It's terrible because it got a huge charge transfer resistance. The second terrible I can identify is the K2 because it does have a charge transfer resistance, but the good thing is it have a much larger linear region, which suggests it does have some capacitance performance. But if you can see the K4 and K3, they are much better because they have less charge transfer resistance, and they have a very good capacitive performance. Now, how can it be related to the cyclical timeogram? So you can see here from the cyclical timeogram that the K1 which resemble with this trend. You can see the separation between the oxidation and reduction peak is much more than rest of the cases. And as I mentioned, that for an ideal system, 57 divided by n millivolt should give you the idea of reversibility or not. So in my opinion, the K1 is extremely poor in reversibility, whereas the K4, which is actually attributing to this EIS spectra, I think it is much more better in reversibility in comparison to rest of the electrode material. So yes, EIS and cyclical timeogram can be linked together in terms of the information and the analysis. The last two characterization technique, which are the chronoamperometry and chronopotentiometry, I think they are really important in some fields, specifically in biosensors, electrochemical biosensors, in chemical sensors, uh, and most importantly in terms of hydrogen evolution reaction and oxygen evolution reaction. Why it is important? Because in hydrogen and oxygen evolution reaction, we are actually interested in the electrocatalyst benchmark current density, which is 10 millivolt, 10 milliamp per centimeter square. So what you are actually up to is to provide a specific current, which is the 10 milliamp per centimeter square. And if it sustained that potential for a longer time, then you can actually get an idea of your electrocatalyst, whether or not it is behaving well. In biosensors, why people use chronoamperometry? Because they can actually study the behavior of that sensors if they vary the amount of that specific chemical or the biomaterial. So they are adding it after a couple of seconds or whatever the step they have already decided, and then they just estimate the current response and take a further further analysis of that response, and then they actually deduce the detectivity of that electrochemical biosensor. Whereas in the HER or the OER, it tells you the longevity of that material or durability of that material, that for how long it can sustain that specific current density, and that tells you that whether or not this electrocatalyst is good. The chronopotentiometry is rather similar because In chrono means time, amperometry means current. Here, the similar chrono means time, potentiometry means potential. So you are providing a potential, which potential it is the over potential, and then you sustain it for a longer time, and then you analyze whether or not the electrocatalyst degrade quickly, or it is sustaining its properties for a longer time, and then you can identify whether or not it is good material for HER or OER, whatever application you are dealing with. Another way of I think apart from the HER or OER, some people have reported these technique in the supercapacitor, but I think in supercaps the best way is to run either number of cycle CV cycles or GCD cycle to estimate or to study the longevity of the device or the cells. I think that's all. Thank you very much.
Hello. I think
Yeah.
I got it. Thank you so much, Mr. Faisal. It was a wonderful and an informative session, and I think like people have got like lots of knowledge from this. You explained like really from the introduction to the advancements of like all the techniques we have to learn in electrochemical to understand supercapacitors or like any other electrochemical device that we have to use. It was a nice session, and we will also like upload this in YouTube link. So now we can move on to the question and answer session. And I think like I can read out the questions so that you can answer. Is it okay?
Yes, that's fine. Yeah.
Yeah, okay. Thank you. So we have like lot of questions here. Let me see. The first question. Apparently, the area of working electrode is way too higher than counter electrode. Shouldn't be other way around.
Um, can you repeat the question?
Apparently, the area of the working electrode is way too higher than the counter electrode. Shouldn't we be other way? So always like most of the people are using like high surface area working electrode, rather the current counter electrode will be like very thin or a very small like a wire like platinum wire. So that's what they're
Yeah.
trying to ask, I think.
I think it depends on which application, but in most of the cases, the area of the counter electrode is not considered, whereas the area of the working electrode is highly considered, because that's where your reactions are happening, and that's where your all the phenomena is taking place. So I think the most important is the area of the working electrode rather than the counter electrode, because even if you get a mesh counter electrode, the current response of that of the wire in the mesh electrode, the difference will be very smaller. I might be wrong, but I think the only purpose of the counter electrode is maintaining a constant current between the working electrode and the counter electrode, rather than, yeah.
Okay, thank you. Then the second question, can CV be used to quantify the concentration of an analyte in the electrolyte?
I think people have used in biosensor application when they just inject some bioanalyte in the cell. So apart from the chronoamperometry, there is another one. I've just forgotten the name, but apart from that specific technique, people have also used the cyclic voltammogram to get the idea how much the analyte is contributing to the cell. Because if you run without the analyte and then you run with the analyte, you should be able to exclude that area from with the analyte, and then you will have the idea of how much the analyte is contributing.
Okay, thank you. Then Can CV be used for the calculation of band edge positions of semiconducting material like organic conjugated polymers?
It's off the top of my head. I think I
Okay,
haven't seen any. Yeah, I haven't seen any papers which include such calculations or measurement. Yeah.
okay, that's true. And like, how can we confirm whether adsorption is happening at the electrode or like not during the reaction?
Oh, what
I
is
think
it?
he's trying to say, he's saying like, how can we confirm the adsorption is happening over the electrode? That's what they are trying to ask, I
It's,
think.
if I'm getting it correctly, I think they are they are meant at whether the reaction is happening at the interface or the bulk. So if the electrolytes are only interacting with the electrode material at the interface, you won't get a proper plateaus in the cyclic voltammograms or the GCD. Well, sorry, not proper oxidation and reduction peaks in the cyclic voltammogram and proper plateaus in the GCD. But if they are going inside the material and they are actually doing the Faraday reactions in the bulk of the material, then you will definitely get proper redox peaks and the plateaus in the GCD of that material.
Okay. And next question, like, sir, please give the difference between accumulation time and quiet time in cyclic voltammetry. How to find the optimum accumulation time in CV?
I didn't get that.
Yeah, we neither like. Please give the difference between accumulation time and quiet time in cyclic voltammetry. How to find the optimum accumulation time in CV?
Well, that depends on the type of material, and then you decide what time you are going to provide the material to store specifically. So, for example, if I have a battery graded material, uh, I think it will be proper to provide a slower scan rate, slower accommodation time rather than a faster. Whereas if it is a EDLC, it can still sustain or can operate properly at a higher accommodation time.
Okay. Then we can move for like two more questions, I think. Is it possible to distinguish between reversible and irreversible redox potential? I mean, redox reaction using CV?
Yeah, this is very important, and this is uh, most of the time if you read paper on battery graded, they actually specifically interested in this parameter. So there's an equation which uses 57, that's a benchmark, 57 divided by n, where n shows the number of electrons transfer during a single process or single activity. So if your system is one electron, which means the difference between your peak should be 57 millivolt. If it is lesser or higher than that, then there are some problem with your cell, and then you have to actually incorporate that or identify that problem, which whatever the problem is. Yeah, so that's what the one of the applications or one of the pros of cyclical thermogram, that it can actually identify and tells you that whether your performance or whether your process is reversible or irreversible.
Yeah, that's good. And like the next question, uh, why we take discharge time difference for calculating the specific capacitance instead of charge time? I think from GCD they are asking.
Yeah, because the charge time is the energy you have invested, and then how much the energy is coming towards you. That in that we are interested, not in the energy we have given to it. So that's why we just focused our analysis or calculations on the discharge one rather than charging one.
Yeah, that's exactly correct. And like, yeah, for the final question, we have actually more questions. So remaining questions will be maybe answered like afterwards. The final question from some anonymous person: while opening the window potential, how do we know the potential window has opened properly, where will be the limit in the oxidation side and the reduction side? Is it always the duct shape that determine a proper CV?
Yeah, that's why I've presented the LSV and its application. So nowadays I've seen number of papers accepted very recently. So people are now using LSV as a benchmark or as a defining characterization technique to give you a better idea of what could be the actual or what could be the stable potential window. So if you run a LSV between lower to higher potential range, and the region in which it is parallel to the x-axis, actually that region is a stable region for you to operate your operate the cyclic voltammogram or the GCD within that limit, and there won't be any redox out of between that potential range. And if you exceed that, there will be a lot of uh, redox or the side reactions happening which can destroy your cell. So I think the duct shape is not the only um, alarm or the only indication to stop there.
Okay, thank you. I think we can. We only have like one more questions. The choice of the electrolyte can be influenced by CV or EAS measurement.
Choice of electrolyte depends on your material actually. What type of material you have, then you can decide the choice of material. If there is literature on certain material that the material is behaving stable and it can it can perform appropriately in basic electrolyte, you can go with the basic one. But if you want to actually differentiate between for your material, which whatever the electrolyte is good for you, then you can actually focus on two or three different parameters. For example, uh, one focus should be on the capacitance, second focus should be on the potential drop in the GCD, and third focus should be on the electrolyte resistance in the EIS. So if these three are going align with each other in all of these three electrochemical characterization, then I think anyone should go with that specific electrolyte.
Okay, thank you. I think that's all. Like, if any question is like left out, maybe we will like answer that in future.
Yeah, no worries.
So,
Thank you.
thank you so much, Mr. Mian Muhammed Faisal. It's a wonderful session, and like you have spent your valuable time to answer all the questions of the participant impatiently.